Analytical or diagnostic digest puts your cloning skills to the test. Mix restriction enzymes with your plasmid and run a gel. What will you see? Will there be one band, two bands, three? The number and size of bands is going to tell you about whether your cloning hopefully works. So whether you were to say stick a gene successfully into a plasmid, so into this circular piece of DNA, by using restriction enzymes, which are sequence-specific DNA cutters, you can make it so that if you have one cut site in the plasmid backbone and one plasmid in your insert, now only if your insert is in there will you get two pieces, whereas if the insert wasn't in here, you would just get a single piece. And you can separate these pieces using gel electrophoresis and then look at the pieces. And so let's get into more details.
So the instructions for making a protein are written in DNA in the form of a gene, and then you get an RNA copy of that DNA made, this messenger RNA, mRNA, which gets used by the protein making machinery, the ribosomes, in order to make a protein. We can take a DNA version of this messenger RNA, we call this the cDNA or complementary DNA, and we can stick it into a plasmid vector backbone. So vector, that's just going to be a vehicle because it's going to allow us to get it into the cells and allow the bacteria to host it for us. We call these circular pieces of DNA plasmids, and the bacteria will keep them inside of themselves, but outside of their own DNA. And this is really helpful because it's going to allow the bacteria to make lots of copies of it for us. And then when the bacteria divide, they're going to keep the plasmid with them. And so then sometimes just when we tell them to, we'll be able to let them make the protein for us. And there are other things that we could do with plasmids other than just getting them to make protein, but that's a typical case that you would see. So we want to make sure that we can get this vector, this plasmid, with our insert. So the gene or the cDNA that we put in there, we want to make sure that we get the right thing into the bacteria. When we're doing this cloning process, we can do it with things like restriction cloning, which I'll talk a little bit about, PCR-based methods like SLIC. In all these cases, you're basically piecing together pieces of DNA, and so we call this recombinant DNA. And when you're doing this, things can go wrong, and your insert, so the piece that you want to get into that vector, into that plasmid, isn't going to actually be there. And you don't want that to be in the bacteria, so we need to make sure that we're only selecting the bacteria that have the insert. The problem is that when we talk about like selecting, the selection that we're doing is based, this actual selection the bacteria are doing are based on the vector itself and not on your insert. So typically in our plasmid, in addition to having the insert or thing of interest, we have a gene for an antibiotic resistance product. This means that if the bacteria are grown in the presence of the antibiotic, they'll be able to survive if they have the plasmid because they'll be able to make the resistance product. But if they don't have the plasmid, then they won't be able to survive because they'll be killed by the antibiotic. So in this way, we're able to select for the cells that take in the vector, but not if that vector actually has the insert, because the insert is totally separate from that selection marker. And so we need a way in order to make sure that our insert's actually in there before we go about just like growing and feeding the bacteria that only have the empty plasmid, or that have the parent plasmid or something, the non-change version. So we need a way in order to check that our insert is actually in there. And we actually have a couple ways. The only way to definitively know that the sequence is right is to send it for sequencing. To purify out that plasmid, send it for sequencing. But there are methods that we can use in order to get a good sense of whether the insert might be in there. We can do things like colony PCR and analytical digest in order to check for these. And today I'm going to talk most about the analytical digest, but I'll also briefly mention the colony PCR, some pluses and minuses of the various techniques. So I'll get more into the details of these, especially the restriction enzyme digests. But both of these methods are basically relying on the fact that if you have an insert in there, the products that you get, if you were to either cut the vector or to try to make a copy of a piece containing that insert, well, the product either wouldn't be there if you didn't have that sequence, or the product would be a different size, depending on how you set up your experiment. So you'll get a different number of pieces or you'll get different size pieces depending on whether or not the insert is actually present. And so with PCR, you're making copies of a region, and with the analytical digest, you're actually cutting out a region. A nice thing about colony PCR, I'll mention and we'll talk about later, is that you don't have to purify out the plasmid first, but with the restriction enzyme digest, you are purifying this out first, which is within the prep kit. So we'll get more into that. But first, let's step back and talk about how this analytical restriction digest works, which means that we have to talk a little bit about restriction enzymes. So restriction enzymes, or like site-specific endonucleases, they're these sequence-specific DNA cutters. So they're these enzymes, these reaction helper mediators, speeder-uppers. Basically what they do is they recognize specific DNA sequences and they cut only at that sequence. There's a lot of different restriction enzymes, as you can see by this chart from NEB, New England Biolabs, who basically has like a monopoly almost over these restriction enzymes. So you can buy a ton of different ones, and they recognize different cut sites, so they can be really useful, although some of them are less specific than others. So they'll be more or less useful because we want to make sure that ours are really specific, and so that we don't have like a ton of cut sites and things like that. And I'll get into this details. You might be wondering, why the heck are there so many restriction enzymes? Well, these are actually coming from bacteria. The names of these, the number, this is actually like a Roman numeral and not a letter. So this is ECO R5, not ECO RB. It's not an electric RB. It's the 5th restriction enzyme discovered in like this E. coli R something strain or whatever. Learned that the hard and embarrassing way when I said ECO RB, and it was like, that's a 5. So yeah, so then that would be 1, etc. And you can see that these are coming from different bacteria. So bacteria use restriction enzymes as part of a restriction enzyme methyltransferase system, where basically what they do is they add methyls, they methylate their own DNA. So methyl is just a CH3 group. They have enzymes that can add on these methyl groups, and this is going to hide the sequences from the restriction enzymes that they have. So this is like, this called this like a restriction modification system, and it allows them to modify their own DNA and then restrict other foreign microbes and viruses and stuff that try to infect them. So there are these viruses called bacteriophages or phages, and they can try to infect bacteria, but the bacteria can then cut them up if those phages have those sequences. And the bacteria, if they have those sequences in their own DNA, they're going to be protected by the methylation. And so sometimes this methylation can actually interfere with when you're trying to cut. There are a few specific ones to keep an eye out for. Like, so DAM is one of the main ones that bacteria use, and this one is actually part of to keep track of which strand has been copied or not. Some of the restriction enzymes overlap with the DAM sites. A few others that you might have to worry about are DCM modified ones and EcoK1. So basically, when you look at those restriction enzyme charts, sometimes you might see things about enzyme or methylation compatibility. Different bacterial strains, though, have different methylation preferences. And so there are things that you can do, and there's typically often an enzyme that you can find that'll fit your needs. And so if you look in any molecular biology or biochemistry lab, you're likely to see lots of these little like ice coolers. These are helpful for keeping these enzymes cold. When you take the boxes out of the freezer, this box stays cold, and you're not having to freeze thaw and stuff a lot. Although these are typically in a lot of glycerol, so they don't like actually freeze solid. You don't want to keep warming them up because that's going to lower their activity. Although I should mention that these things are pretty robust, and so even if it says it's expired, it's probably still okay. But you might want to check with the control just to make sure. As for these numbered tubes, these are like the buffers that go along with it. Traditionally, they've had like individual buffers, but then NEB started making more like universal ones. Okay, so that's the basics of these restriction enzymes and what they were like made for by the bacteria. But we use them for a variety of purposes. One of them is restriction cloning. So we can get the bacteria to cut, we can say we want to move a piece of DNA and insert from one vector to another. We can cut the insert out of one vector, cut the insert out of the other vector, mix and match the pieces that we want. After we run gel electrophoresis, which I'll mention, and then we purify out those pieces. So restriction cloning, that's one place that we use restriction enzymes. And then another key place is when we're doing an analytical digest. So as I mentioned, with these, we're going to be using an agarose gel electrophoresis. Agarose gel electrophoresis is a way that we can separate pieces of DNA by size. Because DNA is negatively charged, thanks to its backbone, it'll be attracted to a positive charge since opposites attract. So if we use electricity, we can set up a positive end charge at one end of a gel, stick our DNA in wells at the other end, and then use electricity to send the DNA swimming towards that positive charge. The longer things are going to get tangled up more as they travel through this gel mesh. So this is an agarose gel, so this is made up of the sugar, and it's this mesh. You can make different percentages if you want to separate bigger or smaller things. So what's going to happen is the less bigger things, the longer things are going to get tangled up more, they're going to travel more slowly. And when you turn off the electricity, then they're going to be higher up in your gel, which you can then visualize with like a fluorescent dye. Then you can learn things about like the number of pieces and the size of pieces by comparing them to a ladder. So this is just a set of pieces of DNA of a known size that then you can compare to. And this is going to be helpful because you're going to get a different number of pieces and different sizes of pieces, depending on whether or not that insert is present. And so the number of pieces you get is going to depend on whether or not your sequence was present, because what you're typically doing is you design this so that you have one cut site in the vector and one cut site in the insert. And we'll talk about how this isn't the only way you can do things. You can also have like two in the backbone, two in that vector. But this is typically the way that we do it, is we have one that's specific for the backbone and one that's specific for the insert. If you therefore have that insert, now you're going to get two cut sites, whereas if you don't have the insert, you're only going to get one cut site. So because that whole circular thing, we want to have at least two cut sites. If we were to only have one cut site, so like a plus minus, say, you would still, both of your products would give you a single band. And what's worse is that the bands, you can't really predict the size, because what happens when you have a circular product is that it can kind of, because it's not linear, it's not snaking through the gel, it's kind of moving weirdly through the gel. And you can get products that are like nicked, or where one strand is broken, or products that are super coiled, so it's really, really wound up. And these different states are going to move differently. And so if you have supercoiled plasmid, it's going to move faster than you would expect based on the linearized form. If you have nicked, it's going to be slower because it's kind of big and floppy. And so it's hard to compare whether or not you actually have a linearized product or you're still circularized. So that wouldn't be very helpful. Hence, we have a site that's both in the backbone and in the vector. But we want to make sure that we only have one of those sites in each places. So you want to make sure there aren't multiple sites for that enzyme on your plasmid, or else you'll get lots of little pieces that are hard to analyze. And so there are tools like NAD cutter, which you can use in order to find restriction enzymes that will fit your need. Sometimes you can use like a single restriction enzyme if there's one site of it in your vector and one side of it in your plasmid already, and then you'll still get the different numbers of cut sites. So there are things like that. But typically you would be using, say, two different enzymes, each of which recognizes one different site. Okay, but say your gene doesn't have any unique restriction sites, or at least none that you have matching enzyme for. So you have a few options. One is to buy another restriction enzyme, but as we talked about, so we typically have a ton of these, and people will get them for like one purpose, and then we don't actually need them for other things, so they kind of just sit around. Another option we'll get into is to introduce a silent mutation, which is going to change the DNA sequence to something that will get cut, but without changing the protein sequence. But first, I just want to talk about the idea of using two restriction enzymes that are going to recognize the vector. This is going to, you might think, not be very helpful, because you're going to get the same number of cut sites either way. But it is helpful because what happens is that if your insert is in there, you're going to get a bigger piece for one of the pieces than if your insert is not in there. So if your insert is not in there, then you're going to get a small piece. And if your insert is in there, you're going to get a big piece. And this way, you're able to use the same enzymes with no matter what insert you have in here. And you don't have to worry about that. So if you were to clone a bunch of different things into here, each of these cases, you could still use these same restriction enzymes. Of course, that also means that if your inserts are about the same size and you're cloning out of one and into another, you're cloning one gene out of one and then another gene into that one. If those genes were about the same size, well, now you would get the same looking products, whether or not you're actually had swapped out the genes. So be careful about that, and you'll get better specificity, more information if you use one that's actually specific to the vector. But if you don't have one, or specific to the insert, but if you don't have one, you can use two that are in the vector. And hopefully the sizes of the pieces will be convenient. Going back to that second idea of introducing a silent mutation, this goes to the idea that, or the fact that DNA, the genetic code is what we call degenerate. So how it works is that as the ribosome moves along the messenger RNA, it's reading out in 3-letter chunks called codons. And so 3 RNA letters spell 1 amino acid, so 1 protein letter. But you can see that, so this is the protein letter that this codes for. And you can see that there's multiple of the same spellings of a single amino acid. So all of these are going to spell for serine, say. This means that we can change the sequence of the DNA without changing the protein sequence. And since the restriction enzymes are only looking at that DNA sequence, we're able to change the sequence of the DNA in order to introduce a cut site that wasn't there before, without changing the protein sequence. And so this is going to allow us to either introduce a cut site if we need a cut site, or we can actually remove a cut site if there were going to be too many. There were like multiple cut sites. And so there are tools you can use, like MolBio tools, silent mutator, silent mutagenesis tool. There used to be a tool that I used called WhatCut, but server's been down. I'm not exactly sure what's going on there, but you can check out this MolBio tools. So there are other methods in addition to restriction enzyme analytical digest, things like colony PCR, and I'll get to those in a second. But before I move on, I just want to mention that a version of this RFLP, restriction fragment length polymorphism, this is actually like DNA fingerprinting, was used typically in it was using crime scenes, as well as genetic mapping to try to find the source of the disease, basically relying on the fact that people have these repetitive regions of their DNA, and they'll have different numbers of repeats. And so the sizes of the pieces in between restriction enzyme cut sites in that DNA are going to be different. And therefore you can cut the DNA with restriction enzymes and see how big the pieces were in order to get an idea about whether DNAs are coming from the same source and that sort of thing. So I have a post on that if you want to learn more. I should also mention that this analytical restriction digest can be used for things other than just cloning in plasmids, if you want to just see if a specific sequence is present. But let's get back to the idea of alternatives, so colony PCR. As I mentioned before, with the analytical restriction digest, one of the annoying things about it is that you actually have to go and you have to purify out the plasmid before you cut it. And so this involves doing this thing called a mini prep, where you break open the cells and then you use these little column kits to purify it out. I mean, it's simple, but it's time consuming. And it's just kind of a pain, especially if you're trying to check a lot of colonies. So you might transform, so do your whatever cloning you're doing, then stick the cloned product into the bacteria, plate that onto the bacteria, get them to grow if they have the plasmid. Then you take these colonies, you take individual colonies, you're going to check and see if they have your insert. And so you might have a lot of colonies, and if you're doing a lot of colonies, it's going to be a pain if you have to do a bunch of mini preps only to find out that your plasmid isn't actually in there. And so an alternative that you can do without actually having to purify that out is going to be colony PCR. And with colony PCR, you're basically designing primers, these little pieces of DNA that are going to bookend regions that you want copied. And then you use PCR in order to make copies of the region. And so you can design primers in different ways, either that are in the insert or in the vector, or it's one in the insert, one in the vector, one in the vector, or 2 in the insert, and that sort of thing. And depending on how you design these, you'll get different products in these different cases. So this is nice in that you can kind of just take a little bit of that gloopy colony, stick it in, like boil it, get it to break open, and just use it straight like that. So you can actually just dip your colony in, and then when the PCR runs, it's going to break the cells open, and you'll get the PCR going without having to actually purify anything. But the bad thing is that you're going to have primers that are going to be like specific for your insert. And so it's not just something that you would have lying around in your freezer. So you typically have to actually design and buy the primers. And so that can be an added pain and an added expense. Whereas with the restriction enzymes, you often have what you need just sitting around in the lab. Okay, but either of those techniques, they're not going to be able to tell you if the sequence is actually present. They're just going to be able to tell you whether like something with a similar size is there. To get if the sequence is present and if it's okay, all the spelling's right, you have to do some sort of DNA sequencing. Traditionally, we would use something like Sanger sequencing, which is just like this traditional sequencing method. Nowadays, we're often using like a whole plasmid sequencing where you actually send your whole plasmid for sequencing rather than just giving them the plasmid and primers, telling them where to copy and then to sequence that copy. Instead, you do like the whole plasmid. You want to keep a close eye on where the plasmid intersects with where you like the cloning sites, like where the stitch sites are. That's where often you can get errors and boo-boos and typos and stuff. So that's where you want to really check to make sure it's okay. Should also mention that if you want to get an idea of which colonies might have an insert before actually doing the colony PCR or before doing the restriction digest, there are screening methods. So a selection, that's like what we're doing with the antibiotics. Here, only the good ones could survive. So we're selecting for the ones that we want. With the screening method, well, here we're just saying, okay, well, show me which ones are good and which ones are bad. But here, don't get rid of anything, just show us. And so with the blue-white screening, say, we're able to get the ones that are probably good and to look blue, and the ones that are not good to look white. And so how this does it is we put our insert in a way that's going to disrupt a gene that the bacteria need to make a blue product. And so if we disrupt that, then they're no longer blue and they're going to look white. But this is just telling us if something got in there, not if the right thing got in there. And so to get an idea about if the right thing got in there, that's when we use the colony PCR or the restriction enzyme digest. And then to test if the spelling is okay, well, then that's where we skip the sequencing. But blue-white screening is a way that you can kind of prioritize which of those clones to test. Hope this helped you understand analytical restriction enzyme digest, some alternatives, and happy cloning.